Hi, Sibilakshmi. Can you hear me?
Hi, ma'am.
Hi. So we'll wait for a few more minutes and see if anyone else joins. Otherwise, we'll start discussing. Okay. So hello. Good evening. My name is Ankita. I am one of your TAs for the course on Basics of Biology by NPTEL. As you already know, we will be having a session every Monday from 7 to 9 p.m. And there are also other TAs who are also clarifying doubts and holding live interaction sessions for the same course. So you can join any of them as per your convenience, and you can clarify your doubts or gain any more ideas of any concepts that you need help with. If you're comfortable, I would recommend you also switch on your videos, or you can do so gradually with time, not a problem at all. So let me first tell you what we'll be doing during this sessions. So first of all, we will address any queries that any of the students have and from any part. But keep in mind, we will not be discussing the assignment which is already active. For example, you have your assignments for both week 1 and week 2 due during this, I think, day after tomorrow. So we will not be able to discuss the answers for those assignments. But what instead we'll do is we will look at sample questions which are related to the topic that has been discussed already. And we'll see how you can solve those questions, not just to help you with this assignment, but also for your exam and for other future exams as well. Since this course is of 12 weeks, what we'll do is we'll hold 12 live interaction sessions. And during each week, we will discuss, during each session, we will take up the topics of each week and we'll try to clarify as much as is possible. And at the end of the session, I will also ask you to suggest any inputs you have from your end regarding anything extra you want, any inputs, any text recommendations, or anything you want me to change, and we'll try to accommodate that as well. So for starters, I have a set of sample questions that I'll discuss with you. And also I will ask you to, if you have any queries at any point of time, please feel free to stop me and ask any questions. Do not hesitate. And apart from that, we'll also briefly go over the video lectures for week 1, where if you have already watched the uploaded lectures, Professor Vishal Dwivedi, he has discussed about the classification of living organisms and about biology in general. So even if you belong from a different background, do not worry if you don't have much knowledge regarding biology as well. You can ask and we'll try to address each and every query over there. That is pretty much it. If you have any questions right now, you can go ahead and ask. Okay, if not, I will share my screen. Please let me know if you can see. Is it visible? If you guys could answer, that would be very helpful. Hello, am I audible?
Yes, ma'am.
Okay, you can see my screen as well, right?
Yes, ma'am.
Okay, thank you so much. Okay, so during week one, what we have basically covered is that what is the definition of science, what are some of the major contributions that have been made by some eminent scientists to the field of biology, the extensive diversity that exists in the living world, and how that is bringing about the need for their classification, for their ease of understanding, and for actually categorizing them into different groups. Further, what is the criteria for that classification, for that grouping, and how does it at all help us? Then we have talked about the five kingdom system of classification, which was given by Dr. R.H. Whittaker, and how he established that there would be that the living world in particular can be divided into Monera, Protista, Fungi, Plantae, and Animalia. Further, he has detailed description for the animal kingdom has been provided, whereby he mentioned what are the criteria, and based on those criteria, the animal kingdom has been classified into Porifera, Coelenterata, Ctenophora, Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and of course the chordates. Then the subclassification of chordates, where Urochordata and Cephalochordata, which are relatively considered to be relatively primitive chordates, have also been elaborated, and the subclassification of vertebrates has also been provided. In the final lecture for the first week, we have seen the classification for the plant kingdom, particularly in details about mosses and ferns, and further about gymnosperms as well as angiosperms. So based on what was discussed during the first week, here are some sample problems or sample questions. So the first question is, who is the father of microscopy? If any of you know the answers, please go ahead and share your answer. It can be right, it can be wrong, not an issue. Please go ahead. I can see some people have also joined just now. Please don't hesitate and answer whatever you think is correct. We will then try to figure out how you should approach questions like this in case you're not absolutely sure. Okay, so for people who have joined late, hello, I am Ankita. I am one of your TAs for this course. For today, what we are doing is we are discussing some sample questions for week one, since this is a 12-week course and we will divide it into 12 sessions, 12 live interaction sessions with me and with other TAs for the course. What we have decided for today is that we'll look at sample problems, try to solve them, and overall revise what has been taught during these lectures. And in the upcoming weeks, we will solve the current assignments, the ones which are due on 8th. And if you have any queries from there, we will discuss that in the next session, along with other extra questions. And you can also please provide your inputs on if you want anything differently to be done, or if you need any extra help, any extra text, or if you're facing any difficulty in any part. So please go ahead and share that. And for now, if one of you could at least try to answer this question. So the question right now we are discussing is who is the father of microscopy? Subbalakshmi, would you try to attempt? Whatever answer you feel is correct.
Yeah, ma'am. Option C.
Option C, Louis Pasteur. Okay. Anyone else has anything to counter or agree with her? Yes, Pavitra, what is your answer? Sorry, I could not hear you.
Antina Ben.
Sorry,
Option A, ma'am.
option A. Okay. So
Yes, ma'am.
if anyone else doesn't have any other inputs, uh, the correct answer is Anthony von Leeuwenhoek. He is the father of microscopy. In contrast, what Subbalakshmi said, it was her answer was Louis Pasteur. He is actually not the father of microscopy. What for this purpose, what you can do is you can go back to the lectures. In lecture 1, you will see that the professor has discussed about some of the most eminent scientists who have made very big contributions to the field of biology. And there he has discussed how Anthony von Leeuwenhoek, he actually first identified or discovered protozoa, and he actually called them as animalcules. So while microscopes, particularly compound microscopes, were discovered by a different person who is here mentioned in option B, Zacharias Janssen, the use of that microscope was carried out by Leeuwenhoek, and he was the first person who actually observed and identified organisms using that microscope, and therefore he is given this particular award, you could say, or he's recognized as the father of microscopy. Okay. And if you do not always know answers to questions like this, what you can do is you can, for now, you need to look at some of these scientists. You need to Google what they are particularly good at, or what not good at, what they are identified for, because each of these scientists has made multiple contributions, but one of their contributions is particularly always given more weightage and they're recognized for that. Like, for example, William Harvey, he was a physician or a doctor, but he is particularly recognized for identifying how blood is circulated in the body. He identified the double circulation system, which is how from the heart, blood is pumped throughout the body. So for that, he is recognized in particular. Then Louis Pasteur, he's actually given recognition for a whole different purpose. He was the first scientist who gave the concept of vaccines. So the smallpox vaccine, which smallpox was a disease which had wreaked havoc in the entire world. Everywhere people were dying of smallpox at one point of time. But as you might already know, now it has been completely eradicated or it has been completely removed, and nobody suffers from smallpox anymore. And the reason for that is actually the discovery of vaccines. And Louis Pasteur was the person who made a very huge contribution to that. And it was, you can say, he was the first scientist who actually brought about this concept. And Zacharias Janssen, as I have discovered, he and his son are both credited towards discovering microscopes. They were actually owners of lens or glasses, like spectacles. They own shops like that, and there they actually came up with this idea where you could magnify a small thing and you will be able to see it. And therefore, while they discovered microscopes, they are still not given the credit for being the father of microscopy, as I already discussed. Moving on to the next question. The ribosomal RNA, which is present in both prokaryotes and eukaryotes, is which of the following? So if you remember how prokaryotes and eukaryotes are distinguished, one of the major difference between the two was that their ribosomes, which is the machinery that synthesizes proteins within the cell, that was different between the two. So so if you remember that a ribosome is not just a protein, it is a complex where there are proteins and there are multiple RNAs. So altogether, it is called as a ribonucleoprotein because it has protein and it has RNA, which is ribonucleic acid, right? So if any one of you remembers what is the difference between the two, between prokaryotic and eukaryotic ribosomes, any of you, if you can attempt. Sandeep Gurjar, can you try? Option D, can you unmute yourself and explain why you think that is the answer? Sandeep, hi, can you please answer? Okay, if not Sandeep, anyone else? Okay, if you are not familiar with how ribosomes are different, the answer to this question, sorry, just a second. The answer to this question was correctly given by Sandeep, and it is 5s rRNA. So this is the prokaryotic ribosome and this is the eukaryotic ribosome. The difference between the two, first of all, is that prokaryotic ribosomes are said to be 70S. Eukaryotic is, on the other hand, 80S. Here, S is a unit of measuring the sedimentation coefficient. In case you need notes, please write this down. This will help you. So S denotes Swedberg unit, which is spelled as SVEDBERG, Swedberg, named after a scientist, and that denotes the sedimentation coefficient. Now, what is sedimentation coefficient? It means that if you spin or if you centrifuge a prokaryotic ribosome versus a eukaryotic ribosomes, they will settle down or they will sediment at different speeds. So based on that, we have been allotted these values, which are 70S and 80S respectively. Now the prokaryotic ribosome, like I said, has proteins and it has RNA. So the proteins here are 50S. If you see here, they are 50S and 30S, along with these RNAs, which are labeled as follows. So the 50S subunit is the large ribosomal subunit, and it has 5S rRNA and 23S rRNA. When we write rRNA, it means ribosomal RNA. Okay. The other types of RNA are mRNA, which is messenger RNA, and tRNA, which is transfer RNA. Transfer RNA or tRNA is required during the process of protein synthesis. And the protein is synthesized from the messenger or mRNA. We will get into all of these eventually during the course, so don't worry about that. For now, you can just see that these are the RNAs which make up a prokaryotic ribosome. And if you compare that to the eukaryotic ribosome, here the RNAs, here the 80S ribosome is made up of 60S large subunit and 40S small subunit. And the RNA sedimentation coefficient also differs here. The only point common between the two, or the only rRNA common between the two, is the 5S rRNA, which was correctly answered by Sandeep. Okay. Coming to the next question. The type of association where two organisms function as a single unit and help each other for survival is known as what? So we are talking about the interaction or the association between two separate organisms who are capable of surviving on their own, but they prefer to live together as a combined unit, and that actually helps their survival or increases their chances of survival. So, what is this type of association known as? Somebody please try to answer. You can answer in chat if that is more comfortable, not a problem.
Symbiosis.
Correct. Can you explain what symbiosis is? Don't hesitate, please. Whatever you know, please try to answer, and we can, of course, rectify or agree with it. Okay, no problem. So, as the question already addresses, symbiosis is a type of interaction or an association between two organisms that helps them to survive when they function together as one unit. Now, symbiosis is not just of one type. There are multiple types of symbiosis or symbiotic interactions. And the three major types are mutualism. Mutualism is a condition where two organisms, they both are benefited by interacting with each other. The second type of symbiosis is parasitic, where one organism lives as a parasite. A parasite is something um, which is harming the person it is living on. So there is one host and there is one parasite. So the parasite benefits from living on the host, but the host is adversely affected because the host is losing its nutrients or losing something which is essential to it. And the third type is commensalism, where one partner or one interacting organism is benefited and the other one remains unaffected by that interaction. So these are much more details, but in general, when two organisms function together and help each other for survival, you call that a symbiotic interaction. And there is some issue here. And what you can see here is an interaction between clownfish and the other organism that you see in the background, that is a sea animal. So what happens here is that the clownfish is responsible for feeding on some small invertebrate or small microorganisms that can harm the sea anemone, and therefore it is helping the sea anemone by protecting it from those organisms. On the other hand, because the sea anemone is structured such that it is providing a place for the clownfish to live and to survive, it is giving a shelter for the clownfish while the clownfish is helping in its protection. So both the partners are functioning as one unit and they are helping each other in survival. You might actually have seen pictures or videos like this in some animated movies, etc., where they show this type of interaction, and it actually exists in the living world. You will find a lot more examples. You can go into it and look at it in detail, and it will help you get a better understanding. Uh, if you look at the other options in this question, asymbiosis is not particularly an interaction. Amensalism is a different type of interaction where if you consider two partners, one of the partners is negatively affected or is harmed, and the other partner remains unaffected. So they are not helping each other for survival, and therefore that option would not stand true here. And neutralism is also a condition where neither of the partners is benefited, and therefore it is also not the right option for this question. So that is how you can filter out. So when you are attempting a question that you are not absolutely sure about, you might have read about it somewhere, or you might have some example in your mind. But if you're not sure about the exact answer, what you can always try to do is try to filter out options that don't look correct to you. So you eliminate wrong answers and you try to arrive at the correct one. And what is important is that you need to do this in limited period of time and to do this accurately, not just for this exam, for any other competitive exams or any exams in the future that you might have. So the process of elimination is something you need to get very good at. You'll be able to do that eventually. Anyway, you have 3 months of practice before you actually get to the exam for this course. So it should help you in the long run as well. So coming to the next question. Uh, the smallest unit in a taxonomic classification with fundamental similarities is known as family, phylum, genus, or species. This is a fairly easy question. If any of you would answer.
Species.
Species, correct. Also, can you tell the exact order of classification, starting from kingdom, which is at the topmost? Someone has written in chat. Right, you're correct. Pavitra, can you please go ahead and elaborate? Or anyone else, someone who has not spoken up yet? Anita, can you try to answer? I'll help you, don't worry. The topmost, if you look at the order of classifying living organisms, then at the topmost comes the kingdom, which is what Whitaker has classified the living world into. We have 5 different kingdoms according to his classification. There are other ways also, but that is the most, you can say, most accepted form of classification and the most commonly known. So there we have the 5 kingdoms where there is Monera, Protesta, Fungi, Plantae, and Animalia, as already discussed. After kingdom comes, and then if you try to answer, this is a live interaction session, so your contribution would be very helpful.
Species, ma'am.
Species is the smallest unit. We're talking about the order, so the exact order from kingdom to species. So that will go as kingdom, phylum, class, order, family, genus, and species. So under common circumstances, when we are referring to any organism and you are asked to write its scientific name, you basically go for the binomial classification. Binomial means just the last 2 terms, which is the genus and the species. So for humans, that is obviously written as Homo sapiens. So that is the genus and the species for a human being. If you consider a fruit, mango, then it is Mangifera indica. That is how it is written, and it's the most acceptable form of nomenclature for any living being. So, so by that exact order, you can obviously understand that the correct option would be species for this particular question. So next we come to question 5, which is that the protein-rich layer in the cell body of euglena that gives flexibility to the organism is what? A, the allurone layer, B, the pellicle, C, collagen, and D, S layer. If anyone knows the answer, please go ahead.
Option B.
Option B. Anyone else? Any other answers? Okay, so the correct answer is actually what she has already said, Ruba already said. It is the pellicle. So here we have the structure of euglena, if you see, and the outermost layer for the organism is the pellicle. It is an extra layer which is present in the body of euglena, and that is responsible for giving it the flexibility, which is unique to this particular class. Now, apart from that, other structures also you can see, but with reference to this question, pellicle is obviously the correct answer. Now, in case you don't exactly remember this, if you are to arrive at an answer for this question, what you do is you take each of the options one by one. So, if you look at what is the most familiar or the most common word, if you look at the options for this question, I would guess it is collagen, right? So collagen, you hear about it everywhere. You hear about it in cosmetic products, in skin products, in being responsible for anti-aging and a lot of other contexts, right? So collagen is a more complex protein, which is usually found in higher organisms. Like, for example, in humans, our skin, it needs collagen. So considering that euglena is a more primitive organism, when I say primitive, I mean a less advanced or more simple organism, there you would not, under normal circumstances, find such complex proteins. And so by that process of elimination, collagen would get eliminated. Next, if you think about S layer, then S layer is a unique, you can say a unique protein rich or a unique coating or layer which is found particularly in bacteria. So what happens there is it is sort of the outermost layer which is responsible for unique structures which you will find on the bacterial surface, some proteins, some recognition moieties. When I say moieties, some entities or some units which are very unique to each bacterial species that are present in their S layer, and it is only present in bacteria. So therefore, it does not become, it is obviously the wrong answer or wrong option for this particular question because euglena will not fit into that criteria. Then comes the allurone layer. Now, allurone, if you remember the lecture, the fifth lecture, where Sir had discussed about the plant classification, and he had talked about monocots and dicots and basically advanced plant species, then allurone is a layer which is present right outside the endosperm, and it is unique to plants, and therefore that also will not be the correct answer in this for this question. So by that logic, you come to pellicle as well. And anyway, you should look into the structure because it will help you remember what are the unique features of euglena and it will help you out as well. Now, the organism that lives in extreme salty areas is Halobacterium, Thermoplasma, Pyrococcus, or Sulfolopus.
Halobacterium.
Halobacterium. Anyone else? Nandan, can you answer, please? Oh, he left. Okay, you don't have to leave if you don't want to answer, that's okay, but please try and answer. It is just a live interaction session for your benefit. So Juba, you are again correct. The halobacterium is it belongs to the class of bacteria that live in very salty conditions. So here again, if you look at the other options, you will see that thermoplasma, as the name indicates, if there is thermo in the name, it means it has something to do with temperature. So organisms which can live in very adverse conditions, very bad conditions, where temperature is very high, then that is an example of a situation where where a thermoplasma bacteria or a bacteria they can live under such conditions. Pyrococcus, the next option, is also that group or that genus of bacteria that are able to live under extreme conditions. So you will come across Pyrococcus furiosus, which is a very, you can say, a very important and a very relevant bacteria, which is used, whose protein is used extensively in biological research. You will find it in almost every lab. And the reason for that is that Pyrococcus furiosus, it grows in, it is only exclusively found in volcanoes and in hot springs and areas which are that extreme level of hot. So the reason for that is I mean, scientists, the scientific community has identified that uh, the DNA polymerase of those organisms is very, not DNA polymerase, the polymerase enzymes from these organisms can function at very high temperatures, and we use that for PCR. You might have heard of RT-PCR, particularly because during the pandemic, during COVID, it became very popular. So during PCR, it is another technique, which is polymerase chain reaction. So during PCR, we need very high temperatures of up to about like 70, 75 degrees, very high temperatures. So under those temperatures, our normal proteins are not able to survive and they completely break apart. So you need proteins from these kind of organisms which live under extreme conditions, and therefore it's an example of how understanding organisms and classifying organisms can help you get knowledge regarding how they can be used for our own purpose and for our own research and understanding. So Pyrococcus and thermoplasma are able to live under very high temperatures. And Sulfolobus is also another genus or another group of bacteria that can live in conditions which are very rich in sulfur, mainly in marshy areas. So they are also capable of living in extreme conditions. So altogether, all of these organisms are often called as extremophiles. Extremophiles, if you divide the word, it is extreme and philic. Extreme means extreme conditions, whether it is temperature, pH, salt, or sulfur, or particular any particular nutrient where the organism likes or it prefers to live. When I say phile or philic, it means loving. So salt loving is halophilic. So extremophilic organisms altogether can be used to group all of these different options. So for salty areas, the answer is obviously halobacterium. Uh, Next, question 7. This is also a fairly easy question. So the phylum that is closely related to both chordates and non-chordates and is considered as the connecting link between them is Echinodermata, Arthropoda, Annelida, or Hemichordates. Any of you, please try to answer.
Hemichordata.
Correct. Why do you think so? You can't just give the answer. Please go ahead and try to explain it as well. Okay, no problem. So, Chuba is again right. It is hemichordate. The phylum Hemichordata is considered to be the connecting link. So, if you look at the order of classification within Animalia as well, you will see that right before chordates, this particular class comes in. And as very obviously the name indicates as well, it is hemichordate. So, that is one easy way you could tackle a question like this. If they are asking you for a phylum in case you don't remember. Technically, it is a very direct question, so it should be easy to answer. But in case you don't remember, you will see that all the other phyla, they come before hemichordata. So the phylum which is the closest to chordates, as is indicated by the name and by the order of classification as well, they are considered as chordates. So not chordates, considered to be close to chordates, and therefore they are considered as the connecting link between the non-chordates and chordates. But you must keep in mind they are not true chordata, so they are still classified under non-chordates, just that some of their characteristic features are similar to that of chordates, and therefore they are considered as the connecting link. Do not consider them as chordates. Chordates need to, as the name indicates, chord, which means the notochord, which is the structure unique to the entire group of chordata. The notochord must be present in these organisms at any point of time in their life, which is not true for hemichordata, and therefore you cannot classify them as chordates. So that distinction must be clear in your heads. Next question, the radial symmetry is found in which of the following phyla? Platyhelminthes, Annelida, Cnidaria, and Porifera.
Option C.
Option C, are you sure?
Yes.
Okay, anyone else?
Porifera.
Porifera, anyone else? Okay, so we got two answers, Porifera and option C, Cnidaria. So if you remember the order of classification once again, then Porifera, or as the name indicates, Porifera has pores, right? So it is a phylum where organisms are, they look like they have pores, and you commonly call them as sponges. So if you remember any of the examples or any of the diagrams of these sponges, they do not have any symmetry at all. They can have any random shapes and therefore they are called as asymmetric. So what do we mean by symmetry? We mean that if you consider any particular, if you take the entire organism and you cut that organism in any particular way, the two halves that are generated as a result of that cut, they should be identical or they should be the same, right? So if an organism is asymmetric, if you cut it by any plane, it will not give rise to two identical halves. Compare that to bilateral symmetry. You consider any human being. If you, for example, if you cut me in half, even if I not might not be completely symmetric, but most features like you have two eyes, you have half a nose, and so on and so forth, that makes us bilaterally. All human beings are bilaterally symmetrical. However, since they're looking for radial symmetry, it means that if you cut that organism or if you divide that organism by any of its radii or radius, which means along any plane through the center of that organism, it will give rise to the same or it will produce two identical halves, right? So, Juba, you're correct. It is Nidaria. And here, as an example, I've shown Hydra, which is an example of this particular phylum. And here you can see that if you consider the organism, imagine that there is a sphere, right? Or a circle. Now, for that circle, if this is the center and you cut along any of these planes, it is giving rise to an identical, it is giving rise to two identical halves. So that makes this particular phylum radially symmetrical. Okay. The other options, we've discussed Porifera. The other options are Annelida and Platyhelminthes. Now, Platyhelminthes, as you remember, are flatworms, right? So if you imagine any, for example, if this is a flat surface, right? So if you cut it by any plane, you don't expect it to be radially symmetrical, right? It needs to have a little more dimension. So by that logic, if you remember, Platyhelminthes looks like a plate. That's how you can remember it. It will never have radial symmetry, right? And Annelida, they are also a group of worms, and they are also not radially symmetrical. You only find radial symmetry in a few phyla, so you can keep that in mind. There is Nidaria, there are Ctenophora. Ctenophora, if you remember, they look like jelly, comb jellies. They are known as comb jellies. So they are circular and they have very long appendages, ciliated appendages, and their upper portion, which looks much like a ball, you can think of it as a ball, and therefore they possess radial symmetry. So, and there is also their radial symmetry in mollusca as well. So, in echinodermata as well. So, you need to be a little careful about that. Otherwise, this is mostly the kind of question where you need to memorize a little bit, or if you need help, what you can instead do is you can always try to remember one or two examples, use images, just go to Google, look at the images for each of these phyla and remember those images as examples, and that will help you answer a lot of questions. Even if you cannot recall every single detail, this should be helpful to you, and over time, you will gradually remember everything. Next question. Mollusks use their feeding organ radula to scrape off algae from the rocks. The radula is made up of A. lignin, B. pectin, C. chitin, and D. suberin. Anybody?
Chitin.
Let's go ahead. Sorry?
Option C.
Option C, chitin. And anybody else? Please go ahead. Most of you are very quiet. Anybody else? Suchitra, can you try? Sana has answered. Option C, same answer. Okay. Anybody else? This is a memory-based question. It's just something you have to remember. So, okay, I won't drag this further. You are right, it is chitin. While this is a question that you mostly just have to remember, what you can keep in mind for now is that the other three proteins, particularly if you look at pectin and suberin, these are proteins that are present in the cell wall of plants. Um, Chitin is mostly not found in plants. Chitin is, on the other hand, found in fungi. So the outermost layer or the wall of fungus is usually made up of chitin, and it is often considered as a chitinous exoskeleton. It's a big word. It basically means exoskeleton. Exo means outside. Skeleton means structure, you can maybe say. So the outermost structure or the exoskeleton for fungi is made of chitin. In this particular case, when we are talking about mollusca, if you remember any example of mollusca, say for example, a snail. Snails are mollusks, right? You will see that most of their body, if you remember what a snail looks like, the outer spiral kind of structure is its shell, right? But its actual body is very soft. So it is only some very few structures within the mollusca that are actually made up of something like a hardy material or a strong mineralized material. One of those structures is its feeding organ, which is radula, because its own body is very soft. It is an unsegmented, smooth kind of body which helps it to survive or attach and survive onto surfaces like rocks. Therefore, when it has to scrape off food from such surfaces, as is mentioned in this question, where it is trying to scrape off algae, which is its food source, then it needs to use a feeding organ which is made up of a strong substance. In this case, it is chitin. So that is the correct answer for this question as well. Okay, this is also a very easy question. This you might just know by uh, common sense, by your own observations. So, compound eyes are present in octopus, mosquito, frogs, or humans.
Mosquito.
Mosquito. Anyone else apart from tuber? This is an easy question. You should at least attempt it. Please answer even if your answer is the same. That is also okay. Okay, no problem. It is mosquito, and here you have an image of the way the eye of a mosquito looks when it is observed under a microscope. And as you can see, it is made up of very, very tiny or very minute eyes, and these are individually known as ommatidia, Ommatidia. So ommatids, or in plural, ommatidia, they make up the entire eye of the organism, and these are particularly compound eyes are particularly a feature for very small organisms that have adapted compound eyes as a way of survival. So, do you know of any other example that you see in your everyday life? Any other organism that also has compound eyes? You guys should know this. Okay, now I'll pick names. One of you, please answer. Lavanya, can you answer any other organism with compound eyes? You see it in your homes every day.
Cockroach.
Cockroach. Okay. Are you sure about it? Think about it. Bees, you know, right? Bees are, if you look at any fly or bee, you have flies in your house. Everybody has. They sit on your fruits, etc. So if you even attempt to swat at flies, which you technically shouldn't, they're immediately able to escape, right? You are rarely able to catch flies and trap them in any way whatsoever. So one of the ways in which they're able to tackle such sort of attacks, which can affect their survival, is via compound eyes. So it is an adaptation of many small organisms that are capable of flying away, and mosquitoes are also one example of the same. If you look at the other options, obviously humans don't have compound eyes. Uh, frogs are also, if you looked at frogs, they have two very distinct eyes which are present on either side of their face, unlike us, where both our eyes are able to look at only one direction at one time. Um, Octopus, on the other hand, while they also have simple eyes, their very unique feature, I'm not sure if you've observed before or not, but maybe you can go and check now. Octopus have unique square or rectangular pupils compared to us or frogs, where we have obviously have circular or round pupils. Octopus or octopi, they have square pupils, and that is their unique feature, but they do not have compound eyes. So this is also an easy option, and Tuba addressed it correctly. Okay. The common method of asexual reproduction in Hydra is A, conjugation, B, spore formation, C, binary fission, or D, budding.
budding,
Budding. Anyone else? Please attempt also. Any correct, wrong answers, everything is welcome. You remember what Hydra is, right? I showed the example of Hydra in radial symmetry. Belongs to the group of Nidaria, and it obviously undergoes asexual reproduction, as is shown in this question as well. So, okay, without delaying much, the correct answer is obviously budding. Oh, somebody answered in chat. Option D, yes, both of you are correct, Anita and Sana. The option is correct option is budding indeed. So in the diagram also you can see this is an example of how Hydra undergoes duplication or how it reproduces. So it develops a bud. So Hydra or in particular, Hydra is very capable of uh, very uniquely capable of regeneration, and regeneration is also a process you will learn eventually. But what it essentially does is, from its own body, it is able to produce a smaller bud or a smaller version of its own self. And then it allows that version to grow until it eventually becomes big enough that it can pinch off from the parent body. So if this is the parent body, this is the emerging bud that eventually grows in size, and then it is able to completely pinch off and form a new organism. And when that new budded or daughter hydra becomes capable of reproduction when it matures or becomes capable of reproducing again, it will undergo the same process. So this is a primitive mode of reproducing, and therefore it is found in such lower organisms. When we say lower, we mean organisms that are not as complex, which do not have that much genetic material or that much complexity in terms of processes which are necessary for its survival, and therefore they can undergo such simple methods for reproducing, and therefore to observe such a process in Hydra. Now, here, in case you're not familiar with the other options which are given, I'll briefly discuss those as well. So, have you heard of conjugation at all? Do you? It's a method of reproduction as well, but have you heard of it? Do you know what types of what classes of organisms undergo this process of conjugation? Okay, in case you haven't, conjugation is a process by which bacterial cells they divide. So while bacteria also undergo fission or they divide from one into two, uh, the process by which they actually transfer their genetic material. So genetic material, by that I mean their DNA, right? So their DNA, it's of a very special type, which you will also learn about eventually. So their DNA does not if you consider a human cell or any other higher organism cell, what happens there is if you have one copy of DNA, that DNA is made into two copies, and then each of these copies goes into the daughter cells. In case of bacteria, this does not happen, and instead, that DNA, instead of getting copied, it is transferred or it is shared between two different bacterial cells. And that entire process is known as conjugation. So it's a very detailed process. You don't need to know so much in depth right now. But if you find it interesting, please go ahead. And if you want me to discuss anything in the future, we can do that in the upcoming classes. So that is conjugation, which is seen in bacteria. Then comes the process of spore formation. So spores are formed by a very wide array of organisms or very different types of organisms. Like, for example, if you think about flowering plants, right? You hear about the process of um uh, pollination and how spores are exchanged. So there you can consider that as a spore, but it is not always necessarily so. But apart from plants, also you see spores in case of mushrooms, you see it in case of ferns. So spores are also the method of reproducing using spores is also a primitive method, and but it is found in more advanced organisms than Hydra, and Hydra therefore undergoes budding. The third option coming in this question is binary fission, which, if you just look at the name, it means binary means one dividing into two, or simply binary means two, right? And fission means division, and obviously fusion means the compilation or the fusion, as the name indicates, between two different entities. So binary fission is also seen in primitive organisms, and one example of that is amoeba. If I'm guessing you must have come across amoeba at some point of time, and therefore this is a process which is mostly found in such kind of organisms. So that brings us to the end of this question. Uh, in the next question, the type of organization found in Grantia that helps in the flow of water through the organism is A, gluconoid type, B, Regon type, C, Ascon type, or D, Cycon type. This is a slightly more detailed question, so you can try to answer, otherwise I'll explain it as well.
Cycon type.
No. I accidentally showed the answer, but it's okay. It's the Ascon type. So in case, let me just minimize this. In case you haven't had much exposure to it, so uh, what exactly happens here is that this Ascon, Cycon, Mucon, etc. These are mechanisms, or for example, if you look at this particular arrangement, right? So here these cells are arranged in such a manner that the water flow, because these organisms, they take in a lot of water, and then their flow of water is managed, or it's regulated or controlled through the organism in many different ways. It can be regulated in many different ways. So one of those, this particular arrangement is known as the asconoid or ascon type, which is what we are talking about here in Grantia. It is, if you see, it's relatively simple, whereby water enters through these pores and then it is pushed out in the direction of the blue arrow. The second type is Cycon type, and the third type, which looks relatively more complicated, it has a more controlled way, or a less forcefully water is pumped through the system. So that is the leuconoid type, and Regon is also another type of arrangement, but it is very rarely found. So that's how different types of water flow is, different types of mechanisms by which water can flow have been elaborated here. So this question is actually a memory-based question. There's not much you can do about it. It's something you will just have to recall. So you can try to focus, you can try to look at a few examples and try to memorize them. And hopefully that should be good enough to help you. If you want more details of how this arrangement is actually done, or how water is pumped through the system, why it is necessary, etc., you can mention that, and I'll try to incorporate more of those details in my next session. So that's something we can do. Please keep in mind if you have any suggestions or any inputs, anything you want me to do differently, please note them down. I'll ask you at the end of the session, and please feel comfortable to share that. There's no issue whatsoever. The next question is about the coiled tubular duct-like organs which filter and remove wastes from earthworm is A. Gonadal cavity, B. Pericardial cavity, C. Nephridia, or D. Protonephridia. Keep in mind we are talking about earthworms. First, if any of you remember, earthworm belongs to which phylum? Let's first establish that, and that will help you come to the answer for this question. If you want, you can even type here. Annelida. Anyone else? Sana has answered Annelida. No one? Okay, Sana is correct. So it is annelids. So if you think about the question, it is actually also a question that you, or rather a characteristic feature which you have to remember. So the filtering or the waste removal organ, particularly in case of earthworms, in this case, will be nephridia. So you can draw analogy to humans. So in case you have studied the excretory system for humans before, you will remember that nephrons are considered to be the basic unit by which urine is filtered in humans, and it is extremely crucial. So when you talk about kidney function, you might have heard about kidney function tests that someone in your family or somebody you know might actually undergo. So there, this capacity is actually measured, the ability to filter waste, and therefore it is very crucial to the survival of any organism that, irrespective of whatever you are taking in, the waste that is generated in your body must also be removed correctly so that it does not affect you in any bad way, does not affect your health or your survival in any way. So for earthworms, which as Sana has mentioned, they belong to Annelida. That particular organ is Nephridia. And so that's actually a distinguishing feature between Ascomyces and Annelida. So while both of them are considered to be worms, there is this difference between the two, whereby in Ascomyces you have Protonephridia. Proto is usually it's mentioned, it is indicative of some primitive structure that these organisms have not yet evolved such a complicated or such a advanced structure for filtering. So their filtering organ is called as protonephridia, but in case of Annelida, that organ is, of course, the nephridia, as we have discussed. And that gradually moves towards the nephron, which we see in higher complex vertebrates such as ourselves. And here the other two options. So yeah, here you could have some confusion only between nephridia and protonephridia. So that is one point where you have to remember, and you can actually do that by keeping in mind that Annelida are more advanced than Ascomyces, which actually have the protonephridia. That should help clear your confusion, because the other two options obviously don't make sense here. So gonadial cavity, uh, first of all, if you're talking about a cavity, that cannot be a coiled tubular organ, right? It cannot carry out, it can carry out filtering, but it is not a duct-like organ. And gonadial cavity obviously indicates it is somewhere close to the gonads, which are the sexually reproductive or reproductive organs of any organism, and therefore they would not be involved in waste filtering or clearance. And the second option, which is pericardial cavity. Pericardium, as the name indicates, it is surrounding the cardial area, which cardial obviously means the heart, so it surrounds the heart region. there also filtration is not carried out. So you should eliminate options like this. And once you arrive at nephridia and protonephridia, there your ability to remember basic characteristic features would help you, and you should be able to arrive at the answer to your question. Okay, this is a fairly direct question. So a flowerless plant with no true roots but rhizoids and that can produce spores in a small capsule is A. Grass, B. Mosses, C. Ferns, and D. Maize. Please answer.
Option B.
Sorry, who answered?
Option B.
Option B. Okay. Anyone else? Tuba is right again. Option B or mosses are the correct answer for this. So if you remember the last lecture for week 1, where the classification of the plant kingdom was carried out. So broadly, you can classify the plant kingdom into two parts. So there are plants which are capable of flowering, those that do not flower. So among them we have mosses, ferns, gymnosperms, and angiosperms. So the particular criteria which are mentioned here, when we talk about flowerless plants, we have to look at mosses and ferns, right? Among them, ferns, they have true roots. So that's how you distinguish between. So when we initially talked about the need for classification, the need for this is that they are categorized because they are different with respect to some fundamental features, right? So based on that, they have been grouped into different organisms. So if you consider a flowerless plant that does not have true roots, then it is does not have true roots and it has something which are when we say true roots, we mean they are not well defined. So if you think about roots, what do you think? A plant which has actual roots is responsible for, among many functions, it is responsible for two things. One, it is for drawing up water from the soil, and two, it is for allowing the plant to stay sturdy or stay rooted in the ground, right? For its support, for its stability, and for drawing water. So among these many functions, these are two very crucial functions that define that a plant has true roots. When these functions are not being performed, what you will consider is that a plant does not have true roots, but instead it has root-like structures, which are here called as rhizoids. So rhizoids are a feature of mosses, whereas ferns, they do have actual roots, and they are called as, not called as, they do have actual roots, and they are classified on that basis. Additionally, mosses are also capable of producing spores. So we were previously talking about the spore formation capacity as well, right? So mosses are also capable of producing spores and they do so in very tiny capsules and therefore you arrive at an answer to this question. So if you look at the other options here, we have already discussed how mosses and ferns are different. So if you already know the difference between these two, you'll be able to answer. And the other options here would automatically get eliminated because maize and grass are more complex plants. So maize is an example of a monocot, whereas grasses are example of dicots. So monocot and dicot plants, as you might already remember, are angiosperms, right? So they are flowering plants, and therefore they do not fall under this criteria, and they automatically get eliminated. So that's how you eliminate incorrect options and try to arrive at a conclusive answer. This is also a direct question. So the egg-laying mammal with a beak like birds is A. Echidna, B. Duck, C. Kangaroo, and D. King cobra. So uh, very obviously, you can just look at the question and eliminate a few options. So go ahead and answer.
Duck.
Duck, are you sure? Anybody else you want to try? You can write in the chat box if you want. You need to read the question carefully here. Somebody wrote in the chat. D, no, duck is already eliminated. The correct answer is echidna. So if you look at the picture here, this is this particular organism. So that's actually why you need to look at images of organisms, because I think once you look at this, you won't forget that this is an egg-laying mammal. If you look very carefully at its mouth, it has a beak-like structure, right? Which is not something you would normally find in mammals. So that is an important criteria. So mammal is normally, what do you understand by mammals? You understand that mammals, they give birth to young ones, right? They do not lay eggs. That is one of the distinguishing criteria for them, which is why a mammal, otherwise, apart from just giving birth, they are also, oh, by the way, the actual term which is used for animals that lay eggs is that they are oviparous. Ov is for ovum, which is an egg. So oviparous are organisms that give or that lay eggs, and viviparous are animals that actually give birth to young ones. So while most mammals, like 99% of mammals, are viviparous in nature, there are very few exceptions to that. And that's why you need to pay special attention to that and remember what are some of those examples. So one of those is the echidna. So it is a mammal because of all its other features. Every other criteria qualifies it as a mammal. But its unique feature is that it lays eggs. And particularly, if you have to keep in mind here, its unique feature is by its morphology. By just morphology is something that you can see from the outside. If you look at an organism, what you see from outside, one of its unique features is that its mouth is much like a beak, as you see in birds, and therefore that's how you can recognize it. The other option that was considered by a few of you was the duck. Now, ducks are not mammals. That is the major criteria here. They are oviparous in nature, and therefore you do not qualify them as mammals. And obviously, their beak is much like birds, but they are not the correct answer to this question. And like I said, the question here that will directly remove two options for you is obviously the king cobra, which does not have a beak like birds, as you obviously know. And same is the case for kangaroos as well. So this you obviously know because you must have seen them somewhere before. And that is why I am stressing on making sure that when you read about something, try to associate it with an image, with a photo, which will help you keep it in mind. So this is something you can remember. And there are also other examples. So look into it. You might have heard of platypus. So that is also an example of an animal, of a mammal which lays eggs. So look into that. So that's for this question. Next, we come to the correct characteristic feature of heart and blood circulation, which is present in mammals, is what? Keep in mind, this is an easy question. Don't get intimidated by options that look very similar. That is something which will be done in most exams because the objective is to try and confuse you. Um, because they're obviously trying to get students who are able to pay more attention before they answer question. So before you go to the options, always take some time, take a minute and read the question thoroughly. So if here particularly you are asked about mammals, use yourself as a reference, use any other human being as a reference, and then think about it. How is heart and blood circulated in mammals? And that should help you answer the question without any second thoughts. So anybody wants to attempt it?
Option A.
Option A. Okay. Anybody else?
Option D.
Option D. Okay. Okay, somebody has spoken in chat also, or should you wrote D? All right, you're correct, Anand. So option D is the correct answer. So like I said, to avoid any confusion, use read the question thoroughly. The previous answer, which was given as A, is that it's an incomplete three-chambered and double circulation system. That is not true. If you think about humans, we have a complete four-chambered heart with two arteries, two atrium and two ventricles. Therefore, it's a four-chambered heart, and we have a double circulation system, which means that both the heart and our lungs are involved in the complete process of circulating blood through the body. So when we say double circulation, we mean that the deoxygenated or the blue blood, as it is usually represented, the deoxygenated or the dirty blood. It's not dirty, it's just it's not carrying oxygen. That blood is sent to the lungs from the heart. So that is collected from all the parts of the body, sent to the heart sends it to the lung purifies it, adds back oxygen into it, and sends it back to the heart for its circulation into the entire body. So there is a double circulation system. There are two very vital organs of your body which are playing a very key role there. And therefore, that is the system which exists in complex higher organisms such as ourselves. Uh, the other options which you see, they are present in more primitive organisms. If you study the other phyla a little bit more carefully, you will identify that there are organisms that have three-chambered hearts. Some have partial three-chambered hearts, which if you look at Sir's video, the video lectures uploaded for week 1, carefully, you will see that they're called as three prime hearts. So that's how basically what has happened is over the course of time, over the course of evolution, organisms have evolved to retain the kind of features which helps them survive the best. And among them, till now, the four-chambered complete heart with double circulation is the most beneficial, is the most fine fine-tuned or the most complex way in which blood is circulated through the body, and that's why you see it in higher organisms such as mammals, which obviously includes ourselves. Four-chambered heart is also found in aves, which is the phylum that consists of birds. But here, particularly, since you are asked about mammals, that is something you should focus on. Okay, great. Somebody has sent a picture of the heart. Very good. If you want, please look at that for reference. And keep in mind that this is, whenever you're asked a question about mammals, try to think about yourself and then answer the question. It will help you. Right. The type of scales present in Bombay duckfish is. Okay, this is also not a question which can be explained much in detail. It's a very direct question. So the answer for this is cycloid and ctenoid scales. If you look at the classification, which is done for the group of Pisces, which is the group of fish, you will see that there are two types of sub-classifications for that group, where we have contractiles and we have osteichthyes. So for each of these groups, there are among a large number of differences. One very obvious difference is in terms of their scales, body scales within the body of the fish. And based on that, you can remember it easily. And this is also a very direct question. I unfortunately do not have a picture for this. I'll add it if you need it later, but otherwise you can just look it up. But if you look at the picture of Bombay duckfish, it will help you remember this. And they have this particular type of scales, which are cycloid and ctenoid scales. Then the two pairs of limbs in frogs arise from A, the pectoral and ventral region, B, the pelvic and dorsal region, C, only pectoral region, or D, pectoral and pelvic region. So in case you're not familiar, the dorsal and the ventral side, in case anybody has any confusion regarding this, and particularly sharing this because there is often a lot of confusion. And one of the easiest way to remember it is actually what I used to, one of my friends referred it, and that's how I remember it. The ventral side, remember ventral by V, dorsal by V. So the ventral side is, if you imagine that if you're wearing a V-neck t-shirt, your neck of the shirt always lies towards your front. So the side where you will see the V is the ventral side, and your back is always the dorsal side. So that's how you remember that this is ventral, this is dorsal for an organism such as yourself. So if you have this basic idea, and then you come back to the question that you're being asked that the two pair of limbs in frogs arise from where. So I think by this definition, automatically you would be able to assume and sort of accept that you obviously won't have limbs. Limbs means appendages, like right, like hands or feet, which in case of frogs, they will not arise from uh, the front and the back of the organism. They obviously arise from its sides. So the question here is whether, okay, so if we go by that definition, it automatically eliminates option A and option B because you won't have dorsal and ventral in this context. So you only have options C and D remaining. So the question is, if you have ever, I'm sure you might have come across a frog some point in your life. If not, again, go back to photos. They help you in answer so many questions. We just saw so many examples of that, right? So you will see that their limbs don't arise from just one region, right? Their limbs arise from the pectoral and the pelvis, both. So pelvis is around the waist, pectoral is higher up above the waist. So they have their hands from the pectoral, and they're not particularly hands. If you consider frogs, basically they are all legs. But if you relate that again to yourselves, it will help you understand that if you consider your hands, these are your pectoral region, and around your waist would be your pelvic region. So the two pairs of limbs in frogs arise from these two regions, thereby answering the question. Somebody has mentioned something in the chat. No, Anand, unfortunately, that's not correct. So that's how you can tackle a question like this. Again, refer to an image if you need it, if necessary. And if it helps you, try to remember that mnemonic for dorsal and ventral region, because that's a very easy way to remember. And yeah, that's it. So the next question, we just have two more. Some issues. Yeah. This is a very easy question. So, which is the field of science that deals with human behavior in context of mental processes? A, history, B, linguistics, C, psychology, and D, public administration. This is a very easy question. Please answer it.
Option C.
Option C, somebody in the chat has also written. Yeah, option C, both of you are correct. So like you see, some questions are, they're very direct, right? It's there are no confusions about it. Yes, Subhalakshmi, you're also correct. So obviously history. So obviously, look, that's where the very first lecture comes in. So the definition of science, how science can be classified, and from there you have multiple divisions. So here you can directly eliminate all the other options and arrive at psychology. But in case you had other more confusing options, what you should do is you should try and eliminate the other incorrect options and then try to arrive at a conclusion. So here, psychology, something you want to say, Suman Ketkar? Okay. Anyway, so that is the correct option for this question. The last sample question for today is the book that explains how species evolved over time and gradually diverged from each other through a process called as natural selection is, this is also an easy question. A, origin of life, B, origin of species, C, origin of virtue, and D, the selfish gene. Okay, some answers in the chat. Sorry, somebody was speaking.
Option A.
No, Tuba, you're not right. Few other people have answered in the chat. Yeah, Subha Lakshmi, I think. And probably Suchitra as well. Yeah, you guys are correct. It's option B. It's Origin of Species that was written by Charles Darwin, and it actually gave the very basic idea of evolution, which is something that has, I think, been explored in detail in week 2 lectures as well. So don't worry, we will discuss the topics, the assignments, the current and some old sample question as well in the next week. But for now, it's just giving you a transition from the entire domain of classification and going towards the topic of evolution. So the book actually it showed that Darwin he observed that some particular birds they are known as finches. Just remember them as birds. He saw that when a particular group of birds who eventually belonged to the same group for whatever reasons for some natural process they got like if you have a bigger group and from that group a small group of birds they got separated for some reasons, right? XYZ reason. They got separated. He observed that these separated birds, when they did not have access to the same kind of food as they previously did. So suppose they had initially they had access to um, some kind of fish or some kind of seeds or leaves, something of that sort, but they got separated and no longer had access to those kind of food. So over time, what happened over years, it does not happen in a day, it happens over years, whereby to adapt to the environmental conditions that one is living in, these organisms they changed and their beaks, no, they changed. The shape of their beaks they changed and they adapted to the kind of condition that they lived in. And what happens over time is that the organisms which had um, made or which had accommodated these, when I say accommodated, it means that if you have a group of say 50 birds and from those 30 birds have changed the way their beaks are, that is allowing them to eat the food in this new place, what will happen is that those 30 birds will be able to survive. While the other 20 birds who have not adapted and who have not changed based on the surrounding conditions, they will not be able to survive. Why will they not be able to survive? Because they're not able to eat the food which is there. So they don't have nutrition and they will obviously not be able to survive. So what is this process? Why is this happening? The process, Charles Darwin, he named it as the process of natural selection. So it is a way in which nature is selecting the kind of um, changes or the kind of adaptations which is allowing an organism to survive the best. So from that group of 50 birds, it is possible that apart from the 30 that changed, the remaining 20 might have changed in some other way. And that way in which they changed, suppose they had, instead of 2 legs, they started developing 3 legs. Then having 3 legs is not helping them in any way in the consumption of their food. Instead, having a different kind of beak or having a different kind of food eating or food digesting mechanism, that is more helpful. So what is happening is that nature is selecting for those organisms which have the most beneficial criteria, which is allowing them to survive in new conditions. He combined this entire thought process, or you can say this is a hypothesis, as the survival of the fittest, which means the organism which has, or the animal or the species, not necessarily species, the organism which has the most beneficial adaptations or most beneficial changes that allows it to be the most fit will always survive. So that is the theory that was proposed by Charles Darwin, and it is quite well accepted. But obviously, there are a lot of ifs and buts about that as well. But this is the image that you see here it's an example of, it's actually the book, a page from the book or the manuscript that was published by him. And I think you can get online copies as well if you're interested in reading up on it. And it's a very interesting species. It's a not species, not it's a very interesting hypothesis, which will make a lot of sense, and it will show you that the power of observation can often give you very conclusive results. So those were my sample problems for today. If you have any queries, please go ahead, and whatever inputs or suggestions you have, please let me know. I will incorporate them. So any of you guys, please, if you have anything, go ahead. Um, Otherwise, I will summarize all of the topics and then we can conclude the session. Nobody? So let me ask you from my end then. Uh, do any of you need any extra text materials for these topics? Then I will look up for books, or I'll try to recommend you some websites or some PDF, whatever copies are available. I'll try to circulate that for you. So maybe that will help you in some way in case you need it. You can reply in the chat box if that is more comfortable. No? Okay, no issues. Uh, Apart from that, the recording of the sessions will be available. You will probably be circulated the details for that by the NPTEL team, and you can refer to these questions from there if you need it, just for your own studies or for reference or anything whatsoever. Apart from that, the slides will also be uploaded. We will circulate all of the DAs, not just me. We will circulate our presentations to them as well. So you'll find that accessible also. So that will be the work plan. In case you, sorry, in case you have any doubts from the assignments from this week, I'll not be able to discuss them today, but please note them somewhere. You anyway don't have much time before submission, I think 2 more days. So attempt the best you can. The sample questions should give you an idea of how much depth you need to go into or how much detail you need to remember before you submit your assignment. And please be careful that your assignment marks also do decide your final grade for the course. And please try your best to get all of the questions right. If not, obviously you can keep improving. This is a long course and you'll have a lot of time to do that as well. So do that well, because ultimately the final grade that is awarded, it has the assignments also have a percentage in that. So it will eventually just end up benefiting you. So that's from my end. If there are no more doubts, then I will just summarize all of the topics. Obviously, it is very detailed. It has been discussed over 6 lectures. I have my notes here. So I'll just summarize it all for you. I did not make slides for this because you already have a lot of material to go through. So I did not want to add on to the burden. But just to refresh your memory and for you to, in case you have any doubts from one particular lecture, maybe it will help you get back to that much sooner, and then you can quickly submit your assignment and just be done with it. And you can look into the lectures for next week, because I think week 3 assignment is also live. So you can solve that also very soon, and we can get on with it. So in lecture 1, what was discussed was the diversity of organisms. What is science? Obviously, science is the systemic, organized knowledge on any particular subject. Broadly, it was classified into social sciences and the natural sciences. So, social sciences included the interaction of one human with another human being. Natural sciences were to understand natural phenomena and find explanations for that, giving some scientific inputs, right? So under natural sciences, we had, which you obviously very commonly know, there is physics, there is chemistry, and there is biology, not chemistry and biology, what is more elaborately known as physical sciences, natural sciences, and the earth sciences. So physical sciences involve studying the processes which involve non-living things. Natural sciences involve living things, which is biology, and also obviously it includes chemistry. You have to keep in mind that no field can be studied in isolation. All of these independent fields, they work together to make the world as we know it, right? So that's that. Then we discussed, not we, in the lectures, there was some detail about some of the most important scientists who have made very big contributions, like Aristotle, Charles Darwin, Leeuwenhoek, William Harvey, etc. Then the difference between a living organism and a non-living organism, like how a dust particle can also grow to form a huge heap of dust, and that is compared to a single cell which can give rise to a whole organism. So there's a difference between the two, which actually distinguishes a living from a non-living organism. Then, in the first lecture only, it was discussed how within the living world there is so much extensive diversity because there are approximately 8.7 million species, and therefore, if you want to even remotely try and understand all these organisms, you need to have a lot of criteria for classifying. So mainly that included the shape, the size, the kind of activity they do, and where they are located. So even though that was broadly proposed, that's not the best way to study or to classify organisms. And therefore, the more preferred route is to actually go about common and different features between these organisms. And so based on that, mainly we look at the level of complexity. Like for example, Aristotle had initially given some morphological features for classification, but what Whittaker did was he looked at the organization or the like the cellular structure, the body of the organism, how that organism was able to duplicate or reproduce, and uh, how was it deriving nutrition? If it is a plant, it is autotrophic. If it is an animal, it is mostly heterotrophic, which means it is not able to make its own food. It is eating other organisms or consuming other organisms to utilize as food and thereby obtain energy. So that's what he did. And then that gave us all of the different kingdoms by Whittaker's classification. Monera mainly included all the bacteria, which are mainly classified based on their shapes. So we have coccus, bacillus, vibrio, and spidellum. And these are like coccus is a round shape, vibrio is comma shape, rod-shaped bacteria are bacilli, and spidellum are spiral shape. So that's how they are morphologically classified. But if we look in terms of their properties or in terms of their structure, then that gives us archaebacteria and eubacteria. Archaebacteria are more primitive. Eubacteria are more advanced and they're considered as true bacteria. So the properties for each of these were also discussed. Then the second kingdom, which was Protista, were studied. Then they are unicellular eukaryotes, their mode of nutrition, their differences from other groups, all of that was studied. And under Protesta only, we have discussed about euglenoids and how their outermost covering is quite unique. So they are capable of photosynthesis as well. But in particular, we talked about the pellicle layer and how it is useful. Then there were plants and animals, which obviously has been discussed in detail later. And this classification also showed that how it did not include some other organisms like viruses. Viruses which are considered to be intermediate between living and non-living could not be um, clearly demarcated into any one group. And the second, also, it talked about some associations like um, lichens. Lichens are algae and fungi that form a symbiotic relationship. We have talked about symbiotic relationships as well. So they are also not true organisms and they cannot be classified under these five kingdoms, but they do exist and they exist very commonly. So those are some, you can say, shortcomings of this classification, but it is one of the most widely accepted methods. Then this was done in the second lecture till here. In the third lecture, more detail was provided about how organisms are classified. So there was the level of organization, which was that it could be either cellular, seen in porifera or sponges, tissue level, seen in coelenterata, then came organ level, organ system level. So organ level was seen mainly in platyhelminthes, but organ system levels were observed annelida onwards. So organ system meant like a circulatory system, like excretory system, and so on and so forth. Then the second criteria was symmetry. So we discussed symmetry also in details, how there can be either if the organism can be asymmetric, or they can be there can be bilateral symmetry, or there can be radial symmetry, and based on these this morphological character as well, you can classify organisms. So that was seen here. And thirdly, the developmental stage, which meant the number of germ layers, whether it is diploblastic or triploblastic, was also used for classification, which actually gave rise to the concept of coelom. Coelom being the body cavity. So whether it is a acoelomate, which means it does not have a body cavity, or it is pseudocoelomate, which is a false body cavity, or it is coelomate, which is it does has a true coelom, such as in our case, the most advanced, not the most advanced. It is usually found in higher organisms. So that is what was there. Then there was segmentation, whether the body is segmented, which is obviously a criteria very commonly found in arthropoda, which are insects. And then finally, the major criteria for division was notochord. So that distinguished chordates from all of the other non-chordates. So that's a very significant criteria for classification. So yeah, these were the criteria, and then each of these animal phylum or phyla were discussed in details. What are the features of porifera? There are different types of arrangements, like we talked about in the case of Grantia, for example, the system for or the vascular system or the water pumping or water flow system, which could be asconoid or cyconoid or leuconoid or racon types. These are the different types. These are the different subtypes, which you can study in detail if you are interested, or we can bring it up in the next session as well. Then came Coelenterata, their different arrangements. There are different lifestyles as well. They can be polyps, they can be medusa. So that difference as well came in. Then Ctenophora, which, as I have already mentioned, because of the radial symmetry, they are spherical. They have very long ciliated structures. Then came platyhelminthes, which are plate-like or they are flatworms. Example being the tapeworm. Their arrangements, their unique features. Okay, because they're mainly parasitic in nature, they have hooks or suckers or structures like that, which help in adhering or attaching onto the host body. So that is also a unique feature which is necessary for them to survive. Then examples for that would be Taenia solium or Fasciola hepatica, which are common parasites that you often find people around you are often diagnosed with. So that's also there. Then came the Ascomynthus, which are roundworms, and they have a different type of body plan compared to that of Platyhelminthes as well, and they possess protonephridia, whereas the next phylum, which is Annelida, like we have discussed, they possess nephridia, and the example for that is earthworms. So they have the organ system level of organization. They have jointed appendages if necessary, and particularly metameric segmentation was found in them, which is often seen in case of somites. Then came arthropoda, very commonly called as insects, and they form the largest class of living organisms. Their body is mainly divided into a head, thorax, and abdomen, and they're they are mostly associated with some sort of movement like creepy, crawly, these kind of locomotion or movement is usually seen in their case. They are often found to undergo a particular step in their lifestyle, which is known as metamorphosis, where their development from the adult or sorry, from not the adult, from the immature or nascent stage to the adult goes through a process which is known as metamorphosis, goes through multiple stages, and it is not direct. So that's also a feature which is very commonly found in arthropods. And they are often also considered to be economically very relevant because, for example, if you think about silkworms, for example, they're very, very important for human livelihood and for um, producing resources that are essential for us, for humans. Then about mollusca, their structure, their bodies, their unique features, their radula, mantle. We have talked about the radula, how it is made up of chitin, and their body, it consists of a nephridia, but it also has a gonadal cavity and a pericardial cavity. And yeah, then we come to echinodermata, which are, as the name indicates, echinus means they are spiny. So dermata means their skin, so they have spiny skin. So examples for that also, like the starfish is one example for it. And they have that unique water vascular system. So they have tube feet. They push water through their body to move from one location to another. So they have a unique structure called as the madreporite, which is necessary or which is the opening through which they take up water and they are able to carry out movement from one location to another. And then we talked about hemichordata, we have discussed it as well, which is the connecting link between non-chordates and chordates. So they are also very important, but they are relatively less frequently found, or their proportion is quite less. Example, most commonly cited example is the Balanoglossus, as you might have read, and it was there in the slide as well. So it is the connecting link or the transition phylum from non-chordates towards the next huge phylum of chordata, and which have the notochord in their body at some stage of their lives at least. And chordates versus non-chordates also have a few very distinguishing criteria, which you can also find in the lecture notes. Then the three subphyla would be Urochordata, Cephalochordata, and Vertebrata. So while Urochordates and Cephalochordates are less advanced or they are more primitive subphyla. Commonly, Urochordates are known as tunicates, whereas cephalochordates are known as lancelets. They are more fish-like or more primitive group of organisms. The next group, which is vertebrates, that has also got very huge divisions. Mainly, vertebrata can be divided into agnatha and gnatostomata. So agnatha again has subclassifications. You can go through all of that. It's there in details. But what we are particularly interested in is the tetrapods. Tetrapods means they have four legs, right? So under the tetrapods, we have amphibia, reptilia, aves, and mammalia. So amphibia are amphibians which are capable of living on both land and water. Um, reptilia, sorry, amphibia, they are capable of living, but reptilia differ from the fact that they are capable of crawling and they are considered to be truly reptiles. And in these classes or in these subdivisions of tetrapods, what you need to keep in mind is the chamber of the heart because they differ and they are actually one of the major distinguishing features between these groups, which we have also discussed. That how reptiles they are considered to have a three prime heart because it's not truly a three-chambered or four-chambered. And their skin also is different. Like for example, if you consider amphibia, they have more of a glandular skin. Example, we think of frogs, right? Frogs have a more smooth, soft skin with glands on top, right? Compare that to reptilia, which on the other hand are mostly dry skin, like for example, crocodiles. So that's how you would remember they are different. And yeah, so these are also in terms of their RBCs, there are some key distinctions. So you need to keep that in mind as well. Then we come to aves, which is very broadly considered to be the group of birds, but those birds can be flightless birds or they can be birds that are capable of flying as well. They are warm-blooded, which means they are capable of adjusting their body temperature with respect to the surrounding environment, which the previous tetrapods were not able to do, but apes and mammals were able to do. So their exoskeleton is mainly made up of feathers, and they have hollow bones, which, as you obviously can guess, helps them in flying, which reduces their weight and allows them to fly. So that's also a very unique feature for their case. And also another feature which helps in their or which assists their ability to float or to be able to fly is because of their lungs, which have like air sacs inside, which also adds to this ability, and therefore is you can consider it an adaptation to the lifestyle that we choose. So that is the group of aves. And then there are mammals, which obviously, as you commonly know, mammals are the group of living organisms that give birth to young ones. But they also have other features. They have mammary glands. They have teeth, like very distinctive teeth, which are incisors, canines, premolars, and molars. They have body cavities, they have four-chambered hearts, they have double circulation, like we have discussed, red blood, they give rise to young ones with a placenta, like the process of development. It involves the formation of a placenta, which is the covering that separates the mother's body from the child or the fetus developing inside the mother. So that is the example, and there are oviparous exceptions also, like we talked about Echidna platypus, and marsupials also are there. Marsupials example being kangaroo, where development of the young one occurs in a pouch which is attached to the mother's body. So that's that. And in the final lecture, in lecture 5, um, we talked about the classification of plants, which can mainly be divided into aquatic or terrestrial. But if we talk about the plant kingdom, it is divided into flowering or non-flowering. Flowering plants are gymnosperms, angiosperms. Non-flowering are mosses and ferns. So mosses are capable of forming spore formation capsules. They are simple. They do not have any true roots. They have rhizoids in its place, and they do not have any xylem, phloem, etc., which are commonly called as the vascular bundle. And they disperse their seeds via wind, and they are mostly found to be attached to some sort of damp land most of the time. Whereas if you compare that to ferns, they are considered to have rhizomes. So if you look at the order of complexity, then ferns are slightly more complex, not slightly, they are relatively more complex than mosses, and they have polypodia, which are like tiny little structures which form spores, and they are attached at the back of their. So if you consider this a leaf, at the back of their leaf, you will find polypodia. And mostly they are also found in damp and shady places. And compare that to the other group, which is the flowering plants, we have gymnosperms and angiosperms. So gymnosperms, you commonly can think about them as tall evergreen trees. They have like stems that are quite woody in nature, and their name actually comes from the fact that their seeds do not have an outer covering, or they're said to have naked seeds, and they have as a spiny sort of leaves. And compare that to angiosperms, they do have a covering, and based on the covering on the seeds, they are classified into monocots or dicots. So monocots example would be maize, and they like one easy way to actually identify that is by the venation. If you have heard of venation before, like if you look at a leaf of a maize leaf, you will see that it has parallel venation. And if you compare that to dicots, they have reticulate venation. And dicots, one common example, as was also cited in the lecture, was of a gram seed, right? Commonly known as a chola. So you will be able to split it into two. It has two cotyledons, and it has a different sort of venation, reticulate venation. And an example for that is also the sunflower. So with this, we summarize all of the details for the entire week one lectures, and we also discuss some of the questions. So in case you come up with any doubts or any clarification that you want, please note them. We will discuss any queries you have in the next session. And again, please don't hesitate to ask questions. It might help your other fellows, your peers, or it might help me in helping go back to some topics and come up with better answers for you. So the information for the slides and the recordings will be made available to you in case you need it in the future. And also, if you have questions, please you can post them at any time on the discussion forum. It will be addressed by one of the TAs, and we'll try to get back to you with better answers. And with that, I think we can conclude the session for today. Please feel free to post any doubts. And thank you so much for attending this session. And I hope it was helpful to you. So good night, everybody.
Hello.